Hello and welcome, grade 11 life science mindsetters, to today's exam school show on our paper 2 biology paper. How are you doing today? We've got Aslam in the studio.
Lloyd, fine, I'm doing well. It was not too bad. I was just getting confused earlier. I'm looking for grade 12 on the screen when I wanted to open the document. that tells you how confused we're getting with this grade 11 in this slot.
Yeah, guys, so grade 11s, I hope you guys have all tuned in. So if you guys have tuned in, remember we were running that big competition with Be a Bright Spark competition. Get yourselves the Safeway K53 book, or you can buy the app off the Samsung App Store. And all you have to do is buy the book, keep your receipt, then head over onto our Facebook page, and there is a tab on our Facebook page for Win a Chevy Spark Lite. And all you have to do is click on that tab, enter your details, and you have been entered successfully. So guys, before we carry on, we'd like to give a big thank you to the MTN SA Foundation for funding our exam school, as well as letting you know that today's show is endorsed by the Department of Basic Education. So guys, without wasting any more time, let's carry on. Let's get started with today's.
Thank you, Lloyd. Thank you. Welcome, grade 11s. I got to watch what I say. Grade 11s, welcome to you. We're looking at exam questions, paper 2. Remember last week we covered the questions in paper 1. We didn't get through half of them, but you have them as your study notes. You can go through them at your own time. We hope to get through ours today. Starting very quickly with the challenge question. Not much of content being tested here. However, this idea of putting this particular question here is that very many of you have a problem reading a phylogenetic tree or a clade diagram as is put on the screen here. So they're saying, look at this diagram, which you have obviously on your side, and answer the questions. So they're telling you here, so many million years ago, 0, 100, 200, 500, we had the advent of seed-bearing plants. And from there, flowers, etc., and coming to these different trees and plants as we go along. You need to answer questions based on this. The very big dot there indicates a common ancestor. In fact, that is a very easy and giveaway clue. They don't have to put the big dot there. You need to know where those lines meet. That is where the common ancestor comes from. So let's look at the questions. So which number indicates a common ancestor for maize and palm trees? In which era did seed-bearing plants first appear? How many million years ago did the Protea bush first arise? So you look at your first 3 questions are based purely on the diagram. The last one is a biological term. Provide a single term for plants with only one cotyledon. So that one you don't need to look at the diagram to answer. So that's your challenge question. Let's get straight down to the nitty-gritty of your paper 2 items. Your first question says, study the diagram below and answer the questions that follow. And there they give you a lovely diagram. And they're asking you first and foremost, to which kingdom does this organism belong? You must be very careful not to confuse kingdom, phylum, family, order, genus. You need to know all these stuff that you learned in grade 10 on your classification knowledge that you have learned. So you must be careful that you don't give the wrong answer there. So this one here particularly, and we can see that it's a diagram of the bread mold, and the bread mold is belonging to the kingdom fungi. That's straightforward. Provide labels for numbers 1, 2, and 3. As is customary with us, we label first on the diagram. Number 1 is this whole structure here, and that would be the sporangium. And number 2 has been wiped out there. This would be number 2 there, and that is pointing to the spores. These guys here. Number 3 is this part here. That's holding the sporangium up, so therefore we call it the sporangiophore. It's a stalk that holds the sporangium in a favorable position for dispersal of spores. And There's the answers there, sporangium spores and sporangiophore. Name 2 types of infections. Name 2 types of infections that are caused by members of this kingdom in humans. So what infections do these particular organisms cause in humans specifically? Remember, you have learnt the diseases or infections that they cause in humans and in plants. They only want to know here in humans. And there you can talk about yeast infections, especially among females, also among males. Thrush, which is common among males and females. Athlete's foot, both sexes as well. So those are some of the infections that we can talk about. So you got thrush, you got yeast infection, ringworm, mainly among children that play in the sand. The ringworm then infects them, and obviously the treatment for the ringworm especially is long-term. You need to take a course of tablets for a long time to get rid of ringworm. Athlete's foot is also a prolonged treatment. Just remember, in terms of athlete's foot especially, remember when you look at fungi and their favorable conditions, you talk about fungi's favorable conditions in terms of dark, moist, warm conditions. Now, if you have athlete's foot, if you're going to continue wearing thick socks and you don't wipe your feet after you bath or you wash your feet, you leave them moist, you wear your socks on top of that, and then you put them in the shoes, so you have created dark, moist, warm conditions. So you are giving the fungi its favorable conditions. So what should you do? You'd rather be walking in sandals. Obviously, you can't go to work, Lloyd. With sandals, they probably send you back home. You know, like in school, they say, boy, where's your uniform? So in work,
your black school shoes.
that's right, yeah. In work, they can do the same thing if you're working for the wrong boss. So you've got to be careful. But what we're saying is when you're at home, barefoot, give it enough sunlight, etc., and keep it dry. Keep it dry. That's one of the key feet there to do there. Okay? Name 3 beneficial use. Now we spoke about the harms. Now we want to know how are these organisms, the group fungi, how are they beneficial to us? Obviously, we use fungi. For example, we eat mushrooms. So it's a source of food on one end. We use fungi to make cheese. We use fungi for antibiotics, etc. So those are some of the things. Look for the common ones before you try to articulate textbook answers. Things that you know. Mushroom, fungi, we eat it. Cheese, you're eating it. You're looking at a variety of cheese that's made by fungi as well. And obviously, the other stuff that we spoke about there, antibiotics, obviously. Antibiotics, making beer, wine, cheese, yogurt, baking bread, in baking yeast. Remember, yeast is a fungus. So if you remember that, most baking requires some amount of yeast to rise the dough as such, and therefore we have this situation there. Good. Let's move on. 1.2. Still on microorganisms. Antibiotic tablets may be prescribed by a doctor. You go to the doctor, you have a bacterial infection generally, you take an antibiotic course. Patients are always told to finish the course of the tablets, even though they may be feeling better. So in other words, you're taking a tablet, but you get to the stage where you think, you know what, I'm feeling better. What does the doctor say? Don't stop taking those tablets until they are finished, not when you are feeling better. The graph shows what happened to the number of live microorganisms in the body when someone does not finish a course of antibiotic tablets. Okay? So here's the graph. On the x-axis, time. On the y-axis, number of live organisms. They're not giving you numbers on this side. You know what is more and what is less, obviously. So you needed to read that. In this investigation, what is the independent variable? Remember what I said last week, that the independent variable in life science is generally the physical feature, the thing that you can change. Number one, that is actually the more correct explanation. What the investigator can manipulate, can change, is the independent variable. The dependent variable is the thing that the investigator does not have control over. He cannot change that. It is dependent on the independent variable. And in life science, in a graph situation, the independent variable is usually found on the x-axis. So therefore, with all what I said, your answer would have been time. Dependent variable would be the number of live organisms in the body. Suggest a hypothesis for this investigation. What did they want to do? They wanted to see what happens when, what happens to the number of live organisms when somebody doesn't take the antibiotic. So the two variables that are there would be not taking the antibiotic and the number of live offspring. So you need to put that in a hypothesis. And a hypothesis is a prediction. What's going to happen? So what are we doing? What are we changing? We are changing the factor that we're not taking the antibiotic. We can't change the number of microorganisms. We can change whether we're completing that course of antibiotics or not. So you start with that. When the antibiotic treatment is stopped before time, what can happen? Three things can happen. One is that the number of live offspring will increase or decrease, or it can have no effect. And in that lies your answer. What do I mean by that? Let's look at the answer. So we looked at one variable, stopping the antibiotic. There, up to that point. Let's just undo there. Right. Stopping the antibiotic. That's one variable. Second variable, the number of microorganisms. And the relationship between the two, it either increases, it can decrease, or has no effect. Remember, in your hypothesis, you don't have to base your hypothesis on the results. Hypotheses are generally made before we make the experiment, not after. What we do after the experiment is make a conclusion, not a hypothesis. A hypothesis is a prediction. So whether you say increase, whether you say decrease, although this one here is telling you something else, it won't make a difference because that's your prediction. You can predict that Khan United are going to beat Limbada United 5-0 over the weekend. Doesn't mean that that's going to happen. It's your prediction. So that's your hypothesis. Or you can say, I'm going to work this morning. It's raining. I think it's going to take me 2 hours to get to work. Your prediction. It may take you 2 hours. It may take you 3 hours. Maybe the traffic cleared up and takes you an hour. After that, you can review your hypothesis. Remember that. So not only the right and wrong answers are important here, people. We need to learn the skills that are involved. Please. Okay? Provide a suitable heading for this graph. Remember, in any graph or table, you must try to include both variables. Again, I've already identified the variables for you. It's time or stopping or starting an antibiotic course, and on the other side, it's number of live organisms. So you're going to use those two variables in your heading. You don't have to write line graph, by the way. You don't have to write line graph, although we've included it there. You don't have to. You can start there by number of microorganisms, one variable, in the body over time. You can include there when stopping antibiotic treatment, because that's important information as well. Why does a patient usually feel more ill for 2 or 3 days after starting a course of antibiotics? In other words, you go to the doctor, you take these antibiotics. Why do you still feel sick in that time? Why do you not feel well? It's obvious because the microorganisms are still multiplying in your body. They are still there. And lastly, in this particular question, how many more days would the patient have needed to take the antibiotics to kill all the microorganisms in the body? For that, we need to go to the graph, and we need to plot. If this thing had to continue on this decline, there, it would have completed around day 15. At the moment, what happened? We stopped it here by day 11. So we will have to have 15 minus 11. That will give us plus minus 4 days. And with that, I hand you over to Lloyd.
So guys, that's the end of the first section. Take a little breather, take a little breath, and we'll get right back to you after the break. Welcome back, grade 11 Mindsetters, to today's life science exam school. And we're busy with Aslam, and he's busy going over some of the work. So let's carry on straight away. Let's go for it.
Thanks, Lloyd. Guys, just before we move on with the questions, just to give you what I feel is a decent way to learn these sections that you're talking about, especially with the diversity. I would find if you draw a table of sorts, and you write there, for example, fungi, protists, and so on, all the other stuff that you're learning as well. And you have your little headings there in terms of their structure, in terms of their characteristics, etc. So you have one big table with all of these things. And most of you are computer literate, and you have your own computers. And you have internet access as well. You design this on your own computer, and instead of putting words in these columns, you put the structure that you see, and you copy the page. I mean, Google image, if you just type in fungi structure diagram, you're going to get a diagram. And you place it in here, and the characteristics, you put something there as well. Its biological importance, its uses, etc. You put it there. And the same goes for the animals as well. All your different animals as well, you put them that way. And you will see that it's easy. That means you can look at one page instead of looking at 30 pages of textbook. So you're taking your textbook, obviously using your textbook as a tool to design your own learning tool, and you keep that with you all the time. So keep that in mind as you are going through your work. Let's go to question 2. The following diagram is a cross-section through an earthworm. So they're giving you a lovely diagram there, and they're giving you labels. Fortunately for you, you didn't have to label here anything. They've given you the labels. They're asking you the question firstly, to which phylum, notice I told you to be careful about those words, to which phylum does the earthworm belong? And that would obviously be the phylum Annelida. Describe the earthworm with regard to the following features. So they're giving you some headings, and these are the headings in the animal section of your work. When you're drawing your table, these are some of the headings you're going to use in your heading. Symmetry, number of tissue layers, type of digestive system, coelom, etc. You're going to use the same headings as you're going along. So the first one says, what type of symmetry does, but remember what the question says. It doesn't say list, it says describe the earthworm. So first of all, you're going to tell us what type of symmetry. And we all know by now that the earthworm has bilateral symmetry. And bilateral symmetry means you cut an organism through one plane only, and it will yield 2 equal halves. Now, I've seen in one textbook that they say humans are bilateral symmetry. I want you to consider that, and you post your answers on the Facebook page, whether that is true or not. Are humans bilaterally symmetrical? What does that mean again, bilateral symmetry? It means that when we cut the organism, usually through the midpoint, longitudinally, it should yield 2 mirror images. That means exact halves. I want you to tell me whether that is true or not, whether humans can be classified as being bilaterally symmetrical. Second one is number of tissue layers. When we talk about the tissue layers, we talk about from the embryo, the embryonic layers that give rise to this organism. How many living layers? And you'll notice you're getting these words that end in derm. D-E-R-M, derm, that is the living layers that we are talking about. And those are the tissue layers that we are talking about. Any word that does not end in that doesn't belong here. So in terms of the earthworm, we know it has ectoderm, it has an endoderm, and in between it has a mesoderm. So it has 3 of those layers, so we say that it is triploblastic Obviously, an organism that has two would be diploblastic. So triploblastic, why? Because it has ectoderm, mesoderm, and endoderm during embryonic development. The type of digestive system, we only talk about two types here. Either the organism has a blind gut or a through gut. A blind gut means it only has one opening that ends in a blind end, in a dead end. Whereas a through gut means it has a separate opening for intake of food, ingestion, and it has a separate opening to release undigested food, egestion. Remember, releasing undigested food is not excretion, it is egestion. Excretion is the removal from the body of metabolic waste. That means something that has gone through a metabolic process, is in excess, and it is harmful, we get rid of it. That's excretion. Whereas egestion means we've taken in some amount of food, we didn't digest all of it. So all those things that are not digested, we're getting rid of it, egestion, or we call it defecation. I don't want to use the common word there, right? Okay? I'll probably get fired. Okay. Type of digestive system in the earthworm is a through gut. That means it has separate openings for ingestion and egestion. It means it has a mouth on one end of the alimentary canal. The alimentary canal is another name for digestive system, or the digestive canal, and on the other end, it has an anus to get rid of undigested food. So it has two separate openings. Coelom is a liquid-filled body cavity that normally separates the gut, that means the digestive system, from the rest of the body. And an organism that has a coelom is called coelomate. An organism that does not have a coelom is called acoelomate, without a coelom. The earthworm, you would know, is coelomate. It has a fluid-filled cavity separating the gut from the body wall. E says, explain how a through gut and bilateral symmetry are advantages. That should have been. Well, they are. You could have used it both ways. Are advantages to the earthworm, or we could have said some English, or we could have said advantageous. So in this case here, they're both correct. But not you must know the context that you're using it. How are they advantages? That means many advantages. And how it is of an advantage, advantageous. So you must check your English in that one there. So we're teaching you some life science, Lloyd, and some English today. You've got to pay me a little bit more today, Lloyd. You're getting double the bonus. So how are these things? We spoke about them. What is a through gut? It has a separate opening and a separate opening for ingestion, a separate opening for egestion. And bilateral symmetry can cut the organism in two mirror halves through the middle point. How are these things advantageous to the earthworm? Obviously a through gut, because there's separate openings for food intake and for getting rid of undigested waste products. It would mean that these two will not mix. Ingested food will not mix with egested food. That's one big advantage. Okay? The through gut allows for separate openings for ingestion and egestion, and this ensures that food and waste don't mix. Bilateral symmetry, because the organism in this case is remember the earthworm is nocturnal. Secondly, it's blind. So obviously it has a problem with locomotion. Now, bilateral symmetry allows this animal to move in a straight forward movement. It allows this animal to do that because of its paired organs. Okay? The blood vessels shown in a diagram are absent in flatworms. They're saying here they got lots of blood vessels, the earthworm, but in the flatworms, such as planaria, you could have learned others. Why is this so? Why do these flatworms not have a blood system? What is the blood system there for? Blood system is there for transport. Yeah? The blood transports things around the body. So we want to know why do flatworms don't have. The word flat is already telling you something. These worms are flat. And if they are flat, they are dorsiventrally flattened, number one. And because of this, the cells are in close contact. Therefore, diffusion is enough for transport. They do not need a blood system to transport things from one to the other. The cells are in such remember, this flatworms like planaria is not as big as the earthworm. We can even add that here. The flatworms are generally, well, no, we can't add that because some are larger. Okay, but because they are flattened, the cells are in close contact with each other and the surface. And the surface itself is also thin. So diffusion can take place from the environment, from the atmosphere into the surface, and from the surface, from cell to cell to cell, and vice versa, cell, 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 surface and out. So there's no need for a transport system in these organisms. Explain how earthworms are important for agriculture and in ecosystems. So we want to know in agriculture and in ecosystems. Again here, we need to think about how the animals live, and that will guide us in our answer. Starting with the top there, they help to recycle organic material. In other words, as the animal is burrowing through the soil, with its food that it's ingesting, it takes in a lot of soil. This soil goes through the earthworm's alimentary canal and comes out on the other side, from the anus. In this way, the organism help to decompose organic material. They help the other saprophytic organism. Remember, saprophyte decomposes dead, decaying organic matter. And it also helps to increase nutrient availability. Oops. How? By incorporating organic materials into the soil and by unlocking the nutrients held within the dead organisms. What do we mean by this? It's very similar to our recycling story there, that there was some nutrients that were trapped in dead organisms. By the earthworm decomposing them, they are trapping, they are releasing those nutrients back to the environment. So it's in the cycle that we are talking about there. Okay? And they're adding organic material to the soil. Lastly, no, not last term, it also improves the soil quality. Why? Because they aerate the soil as they burrow. They're making air spaces between the soil particles, and this helps to aerate the soil. And in ecological sense, they provide food for predators as they are part of the food chain. So they're an important link in the food chain. Some organisms like birds, etc., prefer to eat earthworms. So that means it's a source of food for those animals. And in that way, they are themselves a primary consumer in its, well, not a primary consumer, saprophyte, but they are eaten by a predator in that way. We're moving on still with some animals. Give the functions of the following structures found. Now notice that diagram came with, that question came with a diagram and related questions. Another way that examiner will ask you questions straightforward like these. Not straightforward in that sense, but only words. They're saying, give the functions of the following structures found in amoeba. The contractile vacuole. The minute you hear contractile vacuole, your head must go buzzing with osmoregulation. Okay, controlling water. Remember, the amoeba lives in fresh water. What happens? Excess water comes into it all the time because of high and low water potential. Now what happens? There's too much of water inside. Imagine a balloon. You leave it by the tap. If the water keeps coming down in there, that balloon can burst. So what do? In the amoeba, what does it do? Remember, it does not have a cell wall. So the water collects in the contractile vacuole, and this water then is released via the contractile vacuole. Pseudopodia, also known as false feet, pseudo-false, podia, foot. And the obviously, from there you know it's locomotion, one, and also phagocytosis, you may have learned in grade 10, where the amoeba, when it wants to take large molecules into its cell, the false feet form around the food, and this food is then absorbed into it becomes part of the cell. And the rest we're not going to talk about here. And then we move on. Will substances diffuse more easily in and out of the body of an amoeba or that of a bird? Now we must understand that the amoeba is a unicellular oops, that's a big undo. That went far, Lloyd.
A love-hate relationship.
You can say that again. Unicellular organism, whereas a bird is a multicellular. Also understand that the amoeba is a microscopic organism. You can hardly see it without a microscope and so on. So obviously, diffusion will take place easier to the amoeba. That's the first thing you have to give in your answer. Which one? You have to make a choice. And the choice was amoeba. Why? It has a larger surface area to volume ratio. Or your reason could be the bird has a smaller surface area to volume ratio. And you could have brought in these other points that we have mentioned. And I think that's a mouthful. With that, back to Lloyd.
So guys, let's take a quick break, but when we come back, we're going to give you the details as to how you can win a Chevrolet Spark L. Don't go away. Welcome back, grade 11 life science mindsetters. So, before the break, I told you about you could have a chance in winning a Chevrolet Spark L, and I'm going to tell you all about that competition right now. So all you guys have to do is go down to your local bookstore and buy yourself the Safeways K53 book, or you can buy the app off the Samsung App Store. Once you have one of these two products, if you have the book, keep your receipt, because that will be your proof of purchase. Then go over onto our Facebook page, like our Facebook page, Learn Extra, as well as the Safeways K53 page and the Chevrolet page. But on our Facebook page, there is a tab called Win a Spark L, and all you have to do is go over onto that tab, enter all your details, and then you can also get an extra entry if you share that competition with your friends. So guys, this competition does end soon. The competition is closing in 10 days on the 15th of November, so get all your entries in, and then we'll be giving away a brand new car to one of you lucky entrants. So guys, without further ado, let's continue with the lesson.
Thank you, Lloyd. You got any tips to win that Spark?
Yes, I don't know, get yourself one of these books, I guess, then you can just have to hold thumbs.
Yeah, but I got my license already, that's the problem.
Ah, yeah, well,
Okay.
you see.
Okay, mindsetters, welcome back and let's move. Grade 11s, I'm hoping that you're with us. You're learning something from what we are sharing with you. Don't forget to post your questions so we can help you. Remember, your questions are more important than the questions we have. We've selected a few questions, but you may have others that are bugging you. Those are important. Remember, if you don't get through here, you can still get your assistance to the the help desk as well. Learn Extra help desk as well. Tabulate 3 differences between angiosperms and pteridophyte. So angiosperms, we talk about the flowering plants. That already is a clue for you. And pteridophytes are the ferns. Okay, and you've also learned about bryophyta in the plants. These are the ones that you've learned, and you've learned one more on this side. I'm going to put it because it belongs more on this side. But anyway, gymnosperms. The cone-bearing plants, the flowering plants, the ferns, and the mosses. Those are the type of plants. Again, with this, you could make your whole table and do your story with it. And then you can add the fungi also here because they belong more on this side because of the cell wall and so on. But remember, there are differences between them as well. So what are these differences? So obviously, we started there. So we'll say that angiosperms are sperm coming from seed-bearing plants. Pteridophyte are spore-bearing plants. It means they reproduce by means of spores. And angiosperms reproduce by means of seeds. Angiosperms, the flowers are the reproductive organs. The seeds are found inside the flower eventually, in the fruit anyway. And the pteridophytes, the leaves bear this organ or the structure called sorus, one sorus, many sori, that has the reproductive structures in it. Okay? And angiosperms do not need water for fertilization, while pteridophytes depend on water for fertilization. If there's no water, fertilization will not take place. The table shows different types of waste and their decomposition times. Now we're moving to environment, that is human impact. So they gave you a whole table there, nylon socks, tin cans, paper, glass, and so on. Explain the term recycling. This is a definition, so we're not going to waste too much time there. Remember, though, two important things. It's a process whereby used material or waste products are recycled to make new product. This part is important. You cannot say whereby used materials are reused. Because if I take a paper that's printed on one side and I use the other side, this is reusing. And we're using something that has been used. But doesn't mean that we have changed it, so that's not recycle. Recycle means it must go through a process where it's broken down and then made to something else again. Okay? Name one type of waste in a table above that can be recycled. Obviously, from the table, you would know it would be the tin can, the paper, and the glass. Explain two advantages of recycling. That should be easy. People collect and sell waste at buyback centers and benefit, therefore. So there's one is they're selling and they're creating jobs. People who collect waste and take it to recycling depots contribute to sustainable use of materials, which is important. This is more important than creating the job. Sustainable use. That means we're not putting pressure on the natural product anymore. We are using what we've used already, and we're making more from there. Recycling saves energy and therefore reduces the amount of energy used to make new products. Because we're starting with a product that we don't have to take out of the earth anymore. We're just using it as it is, so it's easier to work on it. There's others as well, remember that, guys. Provide two negative impacts of landfill sites. Landfill sites are dump sites, as we call them. Okay. First of all, it's unattractive. It doesn't look nice. It's next to your house. You won't want to live there. More than that, but it may pose a health risk due to poisons, toxins, pathogens. That means it's going to attract pests that carry diseases and pollutants that are found there, gases that can be emitted from there, and so on. Draw a pie chart to represent the data in the table above. So we've taken this here, and we need to draw a pie chart. And remember, for drawing a pie chart, you need to carry with you in exam a compass and a protractor. And you must have your calculator because you have to do calculations. It says here, show all working. You must show your working. Oops. Right. Okay. How do we show our working? We're taking all those things there, and we're working them out as percentages first. We're making them out of percent. They are percentages anyway. And we're multiplying by 360. Why? Because the circle, the angle is 360 degrees. And therefore, this way we're getting our degrees there. Sometimes they're not given in a percentage like this. They're given into amounts. You need to change that to a percentage first and then change the percentage to an angle. That's what you got to do there. Okay? Again, to show you, there's the percentage given. Times 360 in each case will give you the angle. Taking your compass, you're going to draw a circle first. After you draw your circle, then you're going to use the protractor to draw these angles. In these angles, you can either label each one right here, or you can do what is done here. You put some funny things in there, and you make a key for it. Okay? Your normal pie chart graphs marks will be given to you as we go along. I love that one. All right. So this is what we are doing here. Okay? Remember, people, you don't have to put this here and waste your time unnecessarily when you could have written here nylon socks, etc. And you can label them there. It's easy for the person to see that. Okay? Remember also, sorry, when we're talking about this one, when the examiner or the marker marks this, they usually mark this with a transparency. In other words, they take a transparency of this pie chart and they place it over yours. Okay? What they do allow is they allow for a deviance of only one pencil width. So you can't take chances with your angles. You must have a protractor. And you must make sure you use a sharp pencil. And you must be accurate when you're drawing that. So don't forget the equipment, protractor, compass. You must have these because if there's a pie chart in exams, you have to have that. You cannot draw it freehand. Right, an investigation was carried out concerning the emission of greenhouse gases in South Africa. The following table contains the data obtained from this investigation. This was taken from last year's common exam in Gauteng. There we're getting that story here, nitrous oxide, methane, and carbon dioxide. You notice that these are the three more important greenhouse gases that we study. Remember, water vapor is also another greenhouse gas as well. Distinguish between the term greenhouse effect and enhanced greenhouse effect. This question was put because people make the mistake of confusing these two things. The greenhouse effect on its own, there's nothing wrong with it. The greenhouse effect is there in the Earth's atmosphere to keep the Earth's atmosphere slightly warmer than it should have been without it. This makes it comfortable for us to live on Earth. So this methane, the carbon dioxide, the nitrous oxide, they are normal levels in the environment. With these normal environmental concentrations, what happens, when this radiation comes into the Earth's atmosphere and it bounces back, before it bounces back, these gases trap some of this radiation, and in that way they trap some of the heat and they make Earth warmer. So this is the greenhouse effect, which is essential. However, the enhanced greenhouse effect is because these gases have increased because of our activities. They're trapping more radiation than is necessary, and therefore that leads to global warming, and that's the one we're worried about. Okay? Greenhouse effect is a process by which the greenhouse gases in their normal concentrations keep the Earth warm. In other words, when they trap and all that story there, whereas the enhanced effect is a result of these gases being abnormally high levels in the atmosphere due to human impact, and therefore this will lead to that other concept called global warming. Notice the greenhouse effect and global warming are not one and the same thing. They are two different things, but the greenhouse effect or the enhanced greenhouse effect, in fact, leads to global warming. And this, in turn, global warming has other consequences, like melting the ice caps and raising the sea levels, etc., water levels causing floods, droughts, etc., etc. So this whole thing comes under the section called atmosphere and climate change. Whereas the one we did before this will come under solid waste disposal in your syllabus. Always try to place where the thing comes from. Name the independent variable for this investigation, type of gas or greenhouse gas. State a possible hypothesis for this investigation. The percentage emission of any one of those gases, nitrous oxide, methane, or carbon dioxide, remember, that is one variable, will be the highest or the lowest. That's the other side of it. Which greenhouse gas has the highest percentage emission in South Africa? Now it was easy. That one you just had to look at the table. It's carbon dioxide, obviously. And list one source of this emission. Where does this excess carbon dioxide come from? That's also easy. We burn a lot of coal. We've got this big problem at the moment where the silo has cracked and all the coal that was in there. So therefore, we're having all these power outages. So burning this coal at power stations and elsewhere. Agriculture, even now where there's coal stove still being used at home. Agriculture, using cattle. Burning of fossil fuels. These are different things that will release more carbon dioxide in the environment. Give two ways in which South Africans can reduce the level of this gas in the atmosphere. Obviously, we must burn less fossil fuels. In other words, we must look for alternate forms of energy, firstly. Save on electricity use. In other words, in our homes, the lights that we're not using, put it off, because if we save on electricity, there'll be less demand on the grid. And if there's less demand on the grid, there will be less burning of coal, and this will lead to less carbon dioxide. Use public transport. So in that way, we are pooling our resources instead of each one being in a car and releasing carbon monoxide also and other gases as well. And we have to burn fuel in that process as well. So those are some of the things that we can look there. Now, guys, we're moving to to the other parts, that means section C of your question. So far we've looked at, on your page, on your lesson notes, you have section A questions. In other words, you have multiple choice questions, you have biological terms, and you have some A only, B only, A and B, none of the above questions as well. That's your section A typical question. They can also give you diagrams with one-word answers in section A. We've looked at, from the first question till the third question now, typical section B questions. Now we are moving towards the section C questions, okay? So it says the first question, in other words, your essay. And remember, this is generally question 4 in your exams. And question 4 is only made of the essay. And the essay is only made out of 20 marks. It's 17 plus 3. 17 for content and 3 for synthesis. You do not have to write anything for synthesis, as this mark is the mark that your teacher gives you because of your presentation of your essay. There are certain criteria. Look at the past papers, look at your memo for June, etc., and so on, and you will get the idea, or look at some matric papers. Explain why gymnosperms and angiosperms are regarded as being better suited to life on land than mosses. So we want to look at gymnosperms and angiosperms. Why are they better suited to live on land? Obviously, we should start with mosses then in our answer. And when we start with mosses, we talk about their aquatic adaptations. Why? Because we're saying that they're not so suited for land. They are more leaning towards water. And why? First of all, the male gametes are motile and require water for fertilization. Water is essential for fertilization. Without water, they can't have fertilization. The gametophyte cannot control water loss as it has no cuticle. Conducting tissue is very simple, so not very effective. And it lacks supporting tissue. So you're first telling us why you think moths are more leaning towards aquatic. Now we're talking about the terrestrial adaptations of gymnosperms. Firstly, again, water is not essential for fertilization. Pollen grains are protected against desiccation by resistant walls. Pollen grains in the gymnosperm have air sacs for wind pollination. Seeds are protected against desiccation. And seeds have wings for wind dispersal. You see those seeds of the gymnosperm. Here's the seed, and that's the wing. You throw it up, it does a helicopter and comes down, easy for dispersal. Leaves are narrow, needle-shaped to reduce water loss. Leaves have a cuticle and sunken stomata to reduce water loss. They have conducting tissue to ensure efficient transport of water, salts, and food. There's a 12 marks in this particular part as well. So you're going to get 12 plus 5 is 17. And then the examiner or the marker is going to give you marks for your story. And there are only 3 areas that you get marks for. And these are the 3. This is not like an essay in history or English or Afrikaans or any of the other languages. It's relevance, logic, and content, comprehensiveness. Relevance, logical sequence, and comprehensiveness. Relevance, did you talk about what we asked you only, or did you talk about other things? If you spoke about anything else, even if you got your 17 marks, if you spoke about something else in the thing, in between you started talking about ferns, for example, or you started talking about reproduction in the moss, which wasn't leaning towards water, then you're going to lose the mark for, it's only 1 mark there, and you will lose the mark. Logical, when you're explaining, can you see how the answer is set out? They spoke about the moss first, then they spoke about it. Or you could have done 1, 1, 1, 1. Take 1 character at a time and go through it. But as long as there's a progression in your answer. Lastly, how complete is your answer? If the question is asking for 2 things or 3 things, did you mention those 2 or 3 things? Or did you do only 1 part of the answer and you left the other parts out? So that's the 3rd mark. So remember that that's where you get your synthesis mark. Next essay, Water is a resource from the environment now. Water is a resource that needs to be preserved in order to guarantee the survival of all living organisms. So long story. Is there a question in it? Nothing. Is there an instruction in it? Nothing. It's giving you information only. But watch what happens. You get a verb. What is the verb? Discuss. Whatever comes after the verb is what you have to do. What must you do? How the availability and quality of water is being threatened. This is a very wide open topic. You can write a textbook just on this part here. But we don't want all that. We want stuff from the syllabus only, and you have those headings. So you have to, remember the headings were not given to you, so you had to give the headings, number one. What are those things that affect the availability and quality of water, and how? Discussing it. You're not just talking, you're not just listing it, you're discussing it. And we're not going to go through all. I'm just going to go through the headings. You know what to discuss there. First of all, your construction of dams. How does it affect the availability of water? Destruction of wetlands. This one here deals with availability and quality. By the way, these sections that we've done now, this essay here and question 3, where there were 2 human impact questions. Remember, the human impact part of your syllabus is carried over for next year. You get tested on the human impact again next year, as well as osmoregulation, salt control from paper 1, and carbon dioxide content, and insulin and glucose. Those sections are gone on for matric. So learn them well so you know them for next year as well. Okay? Then we talk about poor farming methods. And poor farming methods also, it affects availability and quality of water. And you can discuss it detailed there. I'm not going into all the details. Droughts and floods, and how does this affect us? Exotic plantations and depletion of the water table, or you can talk about alien plants here. Boreholes, wastage, this is a big thing, you can write a whole chapter on that also. Industry, mining and agriculture, and lastly, pollution, how these things will be affecting the availability. And remember, availability is, do we have the water or not? Quality of the water, can we use this water or not? But remember, if the water is of a poor quality, then it's not available. You can have tons of water in front. We have the whole ocean. But the ocean water is salt water. We can't drink it. So therefore, although it's available, although it's there, it's not available for consumption. Remember that. You must know the difference. Last question quickly. Describe 4 ways in which the overexploit, this comes from biodiversity now. Overexploitation of indigenous plants impacts on the environment, and also explain 4 appropriate management strategies that we can put into place. And that takes us to shortage of food, etc. I'm not going to discuss this one. I want to go straight to our answer to the challenge question. Which number indicates the common ancestor of maize and palm trees? Again, I told you earlier, when you look at that, look at maize, look at palm, where do they join? They join here by number 3. So the answer is obvious, number 3. Next question. In which era did seed-bearing plants first appear? So here's the seed-bearing plants. Here's the seed-bearing plants. In which year did they come here? They came around that time there, okay? And that would be, oh, sorry, they said in which era. They didn't give you the time. So the era, there's the era written here for you, Paleozoic era. They didn't ask you for time. The next question, yeah. How many million years did the Protea bush first arise? Where's the Protea bush? There's the Protea bush. Follow the line. It appeared in this time here. You've got to go across. Now watch here. 175 is your answer there. You must be careful how you read that. And the last one, provide a single term. That would be monocotyledonous. Guys, I'm hoping I've helped you. Lloyd.
Cool. Thanks a lot, Aslam. So guys, thank you to MTN SA Foundation for bringing us this show, as well as this show being endorsed by the Departments of Basic Education. So go get yourself this K53 book. Straight after the show, there's going to be a short little movie by 21 Icons about Nelson Mandela's life and how warm his home is in Qunu, Eastern Cape. So guys, stay tuned for those that short little 3-minute movie. And I hope you guys enjoyed the show today. And until next time, keep learning.